Hi, I'm Steve Palumbi, and this is Lecture 12 in the Science of the Extreme Life of the Sea. And in Lecture 11, we talked about lifespan in marine organisms and how some whales and sharks and fish live a lot longer than we ever thought they did. In this lecture, we're going to dig into that a little more and then ask just the biological question, why do different animals age at different rates? What can explain that? In biology and in evolutionary theory. And the first part of that is just to notice that fish, mammals, even some plants, very similar kinds of structures, can live very different lifespans. For example, the shortest lifespan we know for any fish is the pygmy goby, lives for only about 2 months. On the other side of that scale are yellow-eye rockfish from the deep coast of California, and they can live to 100 or 120 years. Now, gobies and rockfish are different fish. Maybe they have different physiologies. Maybe they're built differently. We can just look at a fish that's pretty similar to a golden-eye rockfish, same genus in the genus Sebastes, blue rockfish. They don't live anywhere near 120 years. So even within organisms that are very similar to one another, they can have very different lifespans. This is a similar graph for different kinds of rodents, different families of rodents. And what's shown here is the range of maximum longevities for those different families. And they range from below 5 years up into the tens of years. Naked mole rats, a kind of social communal living rodent living in caves and deserts, live decades longer than that. Even our sort of most familiar kind of rodents, rats and mice, which are murid rodents, rats and mice live very different lifespans, but they're built basically out of the same sort of plan. They're not different in how their basic construction is, but they have very different lifespans. Birds too, this is a graph of the percent surviving in cohorts of birds in captivity. Some of them survival dropping off pretty quickly by age of 9 or 10 years. These are peafowls, whereas night herons is this graph on this side. That same drop off in survival is happening over about twice the age than peafowls do. So again, big changes in lifespan of animals that are pretty similar. And in this case, these are animals that are living in captivity under ideal conditions. We know a little bit about kind of some of the patterns of aging, and that has a lot to do with body size. So in this particular graph, we have generation time here, which is the average amount of time between generations, but it also is a good reflection of lifespan of organisms. And then length on the y-axis. And organisms that are larger tend to live longer and have longer generation times. There's a lot of scatter here. It's just that they're across scales of size from 10 microns to 10 meters. There's a pretty regular change in the generation time and the longevity. So what causes the variation that's there in longevity, and how can we begin to make sense out of it? Well, the easiest thing to do is say, well, maybe organisms live longer when they don't die so fast. So what is mortality rate, and how does that figure into the average lifespan of organisms? And at some level, that makes perfect sense. Organisms that are subjected to a higher mortality rate are going to have lower lifespans. How can we begin to look at the way mortality rate and lifespan are related to one another? And to do that, we can generate a really simple model and just say, well, given a certain mortality rate, or we'll flip it around and say a survival rate from year to year, how many of or a set of organisms that were born, say one year, are living the next year, and the next year, and the next year. So the number of organisms in this cohort at time t, that we're going to call nt, and that's the number that existed at time t minus 1, that is the year before, times the survival rate. And this can be done per day or per year or whatever. And so NT equals NT minus 1 times the survival rate. And then if we want to stack these up and say the number that were around at time 0, that's the initial cohort size, times survival rate raised to the power, that is the number of days in our study, that's how many are surviving to that number of days now. And when you look at that kind of curve, that kind of equation, it generates this exponentially declining curve in this particular case, where you get very low survival after day, about 300 or so. So the reason to do this is that we can look at the relationship between survival here and the changes in, I'm sorry, you can look at the changes in the number that have survived with the survival rate. And small changes in the survival rate have result in big changes in the number of organisms that have survived over time. So this graph looks at mortality rate at 0.1% per day. So survival here is 99.9% per day. This is 1% mortality. This is 2% mortality. And you can see that over time, these small changes in mortality result in very big differences in the number of individuals that are surviving in that original cohort. They all start out the same, but by day 100, there's vast differences in the number left alive with small changes in the mortality rate. So that gives us a handle on the kinds of changes in the external environment that might change how long organisms actually tend to live in their natural environment. But when we dig into this, the next phase, we discover that it's not just that the organisms are only living through a certain period of time because of external mortality rate. It turns out that organisms are also senescing. That is, they're aging over time. They're not staying exactly the same from year to year to year or day to day to day. Their physiology is changing. So senescence is the gradual decay in physiological function that organisms have with increasing age. And in particular, as organisms age, certain kinds of physiological functions begin to decline the most quickly. In the immune system function in vertebrates, for example, declines with age. Repair processes, how fast your skin can regrow, for example, over age. Reproductive capacity all decline with age. We know that this happens in a steady manner in animal populations because this kind of senescence happens even in animals that are living in captivity under very ideal conditions. And we know that the rate of that senescence is different from one organism to the next. So what's the relationship between mortality and senescence? There's been a number of studies that have shown that they're actually pretty well related to one another. This is a set of data compiled by Robert Rickliffs, who was able to show with this that if you look at the baseline mortality rate in a population in the wild, the higher the mortality rate, then the higher the rate of senescence of that same species when you have it in captivity. And that means that the mortality rate and the senescence rate are related to one another, such that organisms that naturally see a lower mortality rate and therefore tend to live longer in the wild have a lower rate of senescence in captivity. In this second part of the lecture on the theory of aging, we're going to focus on senescence as a physiological process. But we're also going to ask, well, what causes it? And then the real question is, how does evolution work on physiology? And why hasn't evolution eliminated senescence? Because it seems like it's not very good for an individual or a species to senesce. So there have been 4 major kinds of arguments for why senescence occurs in animals and in plants. And it occurs in all animals that have been studied to date, although at the end of the lecture we'll pop up with some information about some animals that hadn't been studied so much before that may actually break the rule. Well, the first kind of thing to talk about is material wear out. Maybe it's just not possible to build an animal that lasts forever, that the components of those animals, the bones, the muscles, the nerves, eventually wear out. And so that is what senescence is a gradual wearing out of the components of the body. Maybe it's just important for species to get old, individuals to get old and die to make room for the next generation, and so that younger organisms can come in and then take over and use the space or the resources. But then there's two kinds of more evolutionarily based arguments for the evolution of senescence. Maybe as a sort of surprise, there isn't any natural selection. There's no evolutionary pressure for longer life in organisms. That our natural sense that evolution should basically lead to immortality, maybe there's something wrong with that theory. And then the other evolutionary theory of senescence is that, yeah, there's selection for longer life, but there's trade-offs too, that if you have longer life, then you have to give up something else earlier in life, and maybe those trade-offs are what define the evolution of senescence. The reason why these particular kind of arguments are so interesting is that when we look at animals that are built pretty similarly, like rats and mice, they have the same kind of materials, but those materials seem to wear out at different rates. The rats and mice senesce at different rates. And so material wear out would have to actually explain why the way you build a rat is much better and wears out less quickly than the way you build a mouse. Well, the material wear out hypothesis basically runs into trouble when we realize that actually most biological structures in animals are not just built at birth and then have to stay there. They're constantly renewed and repaired over life. Skin is renewed and repaired. Bones are renewed and repaired. The blood supply turns over every 90 days in humans. And so biological structures are not just wearing out. What's the difference in, say, rats and mice is that they're being repaired at different rates. And so material wear out is part of the process of senescence, but it's not an inevitability because of what the organisms are built out of. It's actually because they're repairing themselves at different rate, and that they're putting less energy into, by putting less energy into repair, then the structures naturally wear out. Argument about the room for the next generation was popular a long time ago until somebody asked the question, well, room for whose generation? Yours or next generation? Yours or mine? And if you think about it, a set of evolutionary rules that say, well, it would be really good for all the older animals to kind of die out so that the younger generation can go up and take over. The question is from a set of parents, well, if you're dying out, or is it your offspring that are going to inherit your part of that habitat, or is it the offspring of some other parents? So evolutionary pressures to leave room for the next generation don't work very well because you have to answer the question, well, who is going to benefit from that? Is it your own offspring? And if so, then selection will work on that. But if the organisms that are going to benefit from your dying or the offspring of some other sort of pair of parents, then evolution won't favor that. Well, it gets us to these other two explanations that are called the evolutionary theories of aging. One is that there's really no selection for longer life. And two is that there's trade-offs in early versus late reproduction. And then the next section of the lecture, we're going to dive into the evolutionary arguments on both of those and show how evolutionary thinking helps us understand the way that natural selection should work on different parts of a cohort as it ages. So in this part 3 of the lecture on the theory of aging, we're going to delve in a little bit into a cohort over time and followed over time to see how evolutionary pressures might be acting on different parts of the cohort as it ages. And we'll use a test tube experiment that was first developed by Peter Medawar, one of the founding fathers of immunological science. And he talked about it as a test tube, but we'll talk about it as dinner plates in a dorm kitchen. And so imagine that you've got a whole delivery of dinner plates to your kitchen at day 0, and they break at a certain rate, and new ones are delivered weekly. And we can look at the cohort of plates over time, and then you've seen this diagram before. If plates are only breaking at a 0.1% per day, then they'll last a long time. If 2% are breaking every day, then they're not going to make it through the school year without having to be replaced. So the mortality rate changes the decay in the number of plates in this particular setting. So if we take that same graph here and we ask, well, how old are the plates in the dorm? Knowing that their new ones are delivered every week and they have a particular decay over time, then we can estimate, well, what's the average and what's the range of ages of plates at any time in the kitchen. And so this is the different ages, and what we can see is that the number drops from 1,000 down as the age goes up. So in this model, old plates are producing very few offspring in the population, not because they can't, just because there's very few of those old plates in the population to reproduce. Now suppose there's a mutation that causes the plates to become more brittle and have a higher breakage rate at some age X. Well, if that age X is low, say a week or 3 days, then that has a big effect on the reproductive rate of the population, because a lot of the young plates that are reproducing there are affected by that brittleness. Their death rate goes up, the population rate drops. Natural selection would then be expected to work on that X to move it further in along the age of the population, make it a week, 2 weeks, 3 weeks, 5 weeks, 10 weeks, etc., and push it as far back as possible. And the further back it pushes it, the higher X is, the lower the impact that that mutation has on the reproductive rate of the population. So the selection against this mutation, which is sort of the senescence mutation in the plates, selection against it is pretty high if senescence happens early in life. But as senescence happens later in life, then the selection pressure against it gets lower and lower. At some point, X might be so high that very, very, very few plates actually naturally get that old. They break a lot before that. They break much more frequently before that, to the point where very few of them are actually old enough to experience this extra brittleness. Now, suppose you have a whole bunch of mutations for this high brittleness, different kinds, and they occur at different ages X. Selection is going to weed out the ones where X is low. Selection is not going to weed out the ones where X is high, because there's very little evolutionary pressure against it. And what that means is that random mutations in the population that cause senescence are going to be eliminated depending upon when that senescence kicks in. If senescence kicks in really early, and a lot of those individuals would have been alive to experience it, then selection will tend to eliminate that mutation. But selection has very little power to eliminate a mutation that happens, that causes senescence in an organism that's so old that it seldom even appears in the population. And that's one of the evolutionary theories of senescence, then, that natural selection has very low power to eliminate a deleterious mutation when that mutation starts to affect organisms after they're usually dead of other causes. What this predicts is that, in fact, we'll see senescence in populations, but only when we hold those populations to live longer than they usually do. And that, in fact, is one of the strong observations in the theory of senescence in the natural world, is that you seldom see senescent individuals out in a natural population. You do see senescent individuals like this bird example in populations that are held in captivity. They're living longer than they usually would. And senescence tends to kick in when those individuals are living much longer in captivity than they ever would have in the wild. So that means that under this theory, the mutations that are causing these curves to drop, those senescent mutations never would have played a role in the natural environment because those organisms wouldn't have gotten that old to begin with. This fourth part of the lecture on the theory of aging talks about another kind of evolutionary theory that can explain senescence. The one that I just talked about simply looked at the number of individuals that managed to reach a certain age, and because that number is always declining with time, then evolutionary pressures to maintain reproduction at those ages keep getting less and less and less over time. And that means that senescence is perhaps an accumulation of mutations that are keyed to start up in individual organisms that are actually older than they'll ever naturally get in their natural environment. And so evolutionary pressures to prevent those mutations are pretty low. But there's another way of thinking about the evolution of senescence that actually causes senescence to happen earlier and earlier in the population than that. And that's a set of theories that talks about trade-offs between long life and other features of the population, in particular early reproduction in that population. And that's been called the theory of antagonistic pleiotropy by George Williams. What it really means is that an antagonism of fitness or benefits from living long or reproducing early, and how that antagonism plays out evolutionarily. So let's go back to our plate examples. And as before, plates die over time, they get broken, and so there's more younger plates in the population than older plates at any time. But suppose a mutation happens in the plates and it increases their reproductive rate, the rate at which a plate naturally gives rise to other plates in the cupboards. But that mutation to increase reproductive rate comes at a cost of decreasing reproduction later. So that's the trade-off. Early reproduction now, higher reproduction now, lower reproduction later. So natural selection will tend to favor that mutation if the overall rate of reproduction of that individual goes up across its whole expected lifespan. It won't favor that mutation if the average reproduction of that individual over its whole lifespan is expected to go down if that mutation comes in. That actually then says that the mutation to increase reproduction now at the expense of reproduction later depends upon how much it goes up now and how much it goes up later and when that switch occurs. That's essentially means that, say, a gene that cuts off, that increases brittleness of a plate later in life, suppose that gene exists. And as we said, the selection against it is going to be relatively low if that age is high, but there's no selection for it. But if that gene that makes a plate more brittle later in life actually causes reproduction earlier in life to be higher, then there's all of a sudden selection for it. And the antagonism essentially is where selection is pulled towards reproduction early, but earlier and earlier, but long life actually increases reproduction as well. So it's that balancing act that evolution is going to be working with. So the evolution will tend to favor mutations that cause a lot more higher reproduction early in life at the cost of reproduction later, but that depends on how much of a boost you get earlier in life and how much of a decrement there is later. But there's lots of nuances to that, and that is probably how evolution is acting so differently on different kinds of organisms, even within the same groups, like rodents or fish. And that's because reproduction, unlike our plate example, reproduction doesn't stay constant over time. Reproduction rate naturally varies over the course of an organism's life. At the beginning of its life, reproduction is often not really high at all. It takes a while for organisms to grow enough to be reproductively mature. And then in some species, reproduction actually naturally increases as the organism ages, as increases in size. So here's an example for fish. Fish have a growth cycle that's different than mammals or birds. Fish are constantly growing with age. Their growth rate slows as they get older and bigger, but they're continually growing. They're also reproducing continuously after they reach minimum reproductive age. And as they get bigger, their reproductive output goes up a great deal. So this just shows snappers. 30-centimeter snappers might produce 360,000 eggs at a time. That seems like an awful lot of eggs, but a snapper that is just twice as long is producing 10 times as many eggs, 3.4 million. So here you can see that the value of living a little bit longer is actually a huge increase in your reproductive potential. That means that selection for longer life in this particular life history will tend to give you higher and higher levels of reproductive potential. It's possible that selection is going to be for very small size and very short life. But it's also possible that selection could be for longer life, particularly when the number of eggs or the number of offspring per individual goes up that way. There's lots of organisms, especially in the oceans, that are built like this. This is a spawning brain coral. The small little globs there are spawning egg masses, eggs and sperm in gamete bundles. And what you can see is that the gamete bundles are scattered across the whole surface of this colony. A small colony will have relatively small numbers of egg bundles. A big colony have many more bundles that are released. So corals, as colonial organisms, are another kind of critter where reproduction goes up very, very strongly with age and with size. So let's basically, what I want you to do is to take these ideas and sort of think about how they would apply differently to different kinds of organisms. Whales that we talked about can live, say bowhead whales can live up to 100 or 120 years. A female bowhead whale is going to produce an offspring every 2 or 3 years. As she gets bigger, she's not producing more and more and more offspring at the same time. On the other hand, organisms like fish and corals, as they grow bigger, are producing many more offspring, sometimes exponentially more offspring as they grow bigger. So the question really is, what does that do to evolutionary pressures on, say, genes that cause senescence? How does evolution work on a gene that would cause senescence at, say, 100 years of age in a bowhead whale versus a coral? The reason why that's important for these kind of organisms is looking back at these golden corals living on the slopes of Hawaii at 4,270 years of age. Senescence is not kicking in those populations over time. In fact, these represent kinds of organisms, colonial organisms, that in a lot of cases we have no evidence that there is senescence. So that breaks the rule that all organisms have senescence. They eventually wear out. And these kinds of organisms, very little evidence suggests that that applies to them.